Hello everyone, welcome back to Dopa classes. Myself Dr.Dunoonul Shibily on behalf of Dopa. Today we are going to discuss an interesting as well as an important topic in human physiology, that is nothing but mechanism of concentration of urine. Mechanism of concentration of urine. Okay. Before moving on to the topic proper, what I want to highlight is the fact that you already know, if you are trying to get a good medical entrance rank, or even if you are trying to achieve a good marks in your +2 board exam or +1 board exam, what you want to focus is nothing other than your NCERT. This thing already you know, I think, because while considering your last 10 to 11 years of entrance questions, everything, literally 99% of your questions are directly as well as indirectly coming from your NCERT. So what you want to focus is your NCERT book alone. So we Dopa are trying to make you learn the NCERT in the most effective and fruitful way as possible. So if you really want to, or if you really have a dream to achieve a good medical entrance rank as well as achieve good marks in your +2, please be updated with us. So let's begin the concept, that is mechanism of concentration of urine. This particular topic is discussed in the chapter excretory products and their elimination, in the heading mechanism of concentration of the filtrate. So let's begin. So students, before moving on to the topic proper, we should have a basic idea on the concept of what osmolarity. Osmolarity. What do you mean by osmolarity actually? This osmolarity is a term which denotes the concentration of solutes in a solution. So osmolarity simply denotes concentration of solutes. Hope you know what is solutes. Solutes in a particular solution. So the component of a solution, we can say like component of a solution have what? Solution have a solute as well as a solvent, and solute is simply, we can say solute is the solid substance present in the solution, and solvent is simply what? Solvent is simply the liquid part of the solution. Usually it will be water. So this much thing we know. So now I am going to consider two systems. Suppose this is system number A, which is a solution, and this is system number B, which is again next solution. Now I hope you can understand. This is what this green thing is actually semi-permeable membrane, which actually permits the flow of water molecules according to the concentration gradient. So while considering the A and B, these red particles which I have added here, these red particles is actually what solutes. So that solutes are added in that particular solution. Here there is more red particle. So I will ask you a question. Which solution, whether the solution A or solution B is having low osmolarity? Osmolarity, hope you know, that is the particular terminology which denotes the concentration of solutes. So I can say here, you can simply say here, what is the osmolarity with respect to A, whether it is high or low. In A, it is actually low osmolarity, because you can see very less red dots there. That means very less solutes are present. So you can just say here there is less solutes are present. Less solutes mean we can say there is less concentration of solutes, or simply we can say like low osmolarity. Now considering the solution B, here there is more solutes are present. More solute means very high osmolarity is present. Now I hope you know this is a semi-permeable membrane which is present in between, that allows the flow of water molecules. So what will be the net direction of the flow of water molecule here? It is obvious. Obviously, it will be from A to B. It will be A to B. I hope you know that particular concept, because the simple explanation given to this is, so we can say the flow will be happening from A to B, right? Flow will be happening from A to B. Why this is happening? The concept is actually the water molecule actually possess kinetic energy actually. So here there is, in solution A there is more amount of water molecule, here there is less amount of water molecule. So obviously the flow will be happening from A to B. So you can say the flow will be happening from low osmolarity to high osmolarity. So low osmolarity to high osmolarity, that is the net direction of the flow of water molecule. Okay, I hope you got that particular concept. This concept you need to have before we are entering to the particular topic, that is the mechanism of concentration of urine. Now we are almost set to discuss the mechanism. So students, now we can discuss the mechanism of concentration of filtrate, or simply mechanism of concentration of urine. So if it is asked like a 1 mark question, if it is asked what is the mechanism of concentration of urine, you can just write it is simply counter current mechanism. Counter current mechanism is the mechanism which actually concentrates our urine, So all we want to do right now is to explain this counter current mechanism happening inside our kidney. So before entering to the topic proper, we have to know what actually is this counter current. Counter current, as it is mentioned, system in which inflow runs parallel to and in close proximity to outflow in the opposite direction. Okay, so suppose let's consider a system like this which have an inflow and outflow, so suppose this is a system which have an inflow and outflow. This is the inflow and this is the outflow. Is this a counter current? Never. Because why? Because here according to the definition, this inflow should be parallel to this outflow and both should be in close proximity. How that is possible? That is possible only by this particular structure. A structure like this, if a structure is like this, it is possible, right? Okay, all right. So this is the inflow and this is the outflow, and this inflow is actually close proximity to this outflow. So this is simply the counter current. This is simply the counter current in which the inflow is actually running parallel as well as close proximity to outflow. That structure, that counter current is actually present in inside our nephron. I hope you know the nephron. Nephron is the structural and functional unit inside the kidney. So see this particular diagram. In this particular diagram, it is clearly visible that our loop of Henle, this is the descending limb of loop of Henle, and this is the ascending limb of loop of Henle. Obviously this can act as a counter current, because here there is inflow which is in the descending limb, and here there is outflow which is in the ascending limb. They are in close proximity as well as they are in parallel. And also this is the vasa recta which is running close proximity to this loop of Henle, and you can see the vasa recta also have an ascending limb like this and a descending limb like this, and also an ascending limb like this. So this is also like the inflow is running close proximity and in parallel to each other. So in effect, the vasa recta as well as the loop of Henle by itself, they form what? They form a counter current. So you can know that what I mentioned is regarding the loop of Henle. The inflow of the flow of the filtrate in the two limbs of Henle's loop is in opposite direction, and thus forms a counter current. So simply we can say this is the descending limb of loop of Henle, and this is the ascending limb of loop of Henle. So see, considering the loop of Henle, the inflow is like this and outflow is like this. They are almost in close proximity as well as they are in opposite direction. And I think you know the peculiar feature of this loop of Henle, the descending limb and ascending limb. One of the peculiar feature, as I already had told in the last session, was this ascending limb of loop of Henle is permeable to electrolytes, and this is permeable to water molecule. This already you know. Now you have to understand regarding this descending limb, but this is impermeable to what? This is impermeable to Na plus, or simply we can say NaCl. Okay, but NaCl, here also we can write NaCl, but here this is impermeable to water. That thing you have to keep in mind. Now, as we already mentioned regarding the vasa recta also forms a counter current. How it forms a counter current? It is because the flow of the blood through the two limbs of vasa recta is also in the counter current pattern. Hope you know the inflow is running close proximity and in parallel to each other. So vasa recta, the two limbs is like this. So vasa recta also have a, here there is it, this was our descending limb and this was our ascending limb. I will represent DL as, the descending limb as DL and ascending limb as AL. So this is the descending limb of vasa recta and this is the ascending limb of vasa recta. Here also a counter current mechanism or counter current flow is happening for the blood actually. So I hope you got. Now what is the permeability thing regarding this vasa recta? Vasa recta, the descending limb as well as the ascending limb, the peculiar feature is that it is the both are permeable to water as well as to water and electrolytes. That thing should be there in your mind. Okay, hope it's fine for you. Now how this ascending limb, how this loop of Henle is arranged, with respect to vasa recta, as I have already shown you in the diagram. In the NCERT has shown like this. It is almost this vasa recta and this loop of Henle are in connection or are in close proximity to each other. That thing also you should keep in mind. I can just represent that like this, just like this I can represent. Okay, they will be like this, just a representation only. Okay, the actual way of this loop of Henle and vasa recta I have shown you in the diagram which is given in your NCERT. So I can just mark now this is the descending limb of loop of Henle, ascending limb of loop of Henle, and this is the descending limb of vasa recta, this is the ascending limb of vasa recta. Hope you got the idea. Now I am going to change the position of vasa recta now. Okay, I am shifting this ascending limb to this direction. So suppose this is our vasa recta and this is the descending limb of vasa recta and this is the ascending limb of vasa recta. Suppose this is the ascending limb of vasa recta. So I have made the descending limb in its position and I have just shifted the ascending limb to this way. So how it will look like? So our loop of Henle is there as itself, but this is the descending limb and this is the ascending limb. Here we have made the descending limb of vasa recta in its position itself, but we have just shifted the position of ascending limb to here. This is the way how we have shifted, because we have to understand, in order to understand the different flow of water molecules as well as electrolytes, we have rearranged this. I hope you got the picture. This is the descending limb, this is the ascending limb of vasa recta, this is the loop of Henle. So in effect both the descending limb are close to each other, both the ascending limb are away from each other. That's the way how they are arranged now. Why I just said that is to make you understand the figure which is given in your NCERT. If you have that thing in mind, only you will get that point. or you will get the point which is mentioned in your NCERT. So this is what? So this is the descending limb of Henle's loop, and this is the ascending limb of Henle's loop. So obviously this is the descending limb of vasa recta, and this is the ascending limb of vasa recta, and this is our Henle's loop. Hope it's fine for you. Now this arrangement you should get clear first. Only after that we will be getting the correct picture. So ascending limb is like this, and descending limb is like this of the loop of Henle, and the descending limb of vasa recta is like this, and ascending limb of vasa recta is like this. So I hope you got the way how this is given in this particular figure, because students always get confused with this figure. They don't know whether this is descending, because that is not mentioned in your NCERT, that you have to keep in mind. Now another thing which is actually visible in the medullary or medullary interstitium is, as we go towards the inner medullary interstitium. Hope you know this is the medulla portion, this is the medulla portion, and this is the outer cortex portion, It is into the medulla actually the loop of Henle is inserted. We have studied the basic difference between juxtamedullary nephron and cortical nephron. There we have told 15% are juxtamedullary nephron, and those nephron were having long loop of Henle, as well as their percentage is only 15%, and you have to understand it is this juxtamedullary nephron which is playing an important role in this mechanism of concentration of urine. So this long loop of Henle is present in which nephron? That is usually in juxtamedullary nephron, and you have to keep in mind this particular nephron also have another feature like this. What is this? This is the thick ascending limb, and this is the thin ascending limb. See here, there is thin ascending limb is present, as well as thick ascending limb is present here. That is a peculiar feature which is found in the usually in the juxtamedullary nephron, which is the main concentration, which is the main nephron which is functioning as the concentrating nephron. Hope it's fine for you. Okay, now we have to understand, on going towards the inner medulla, you see the medullary interstitium. What is the interstitium actually? Interstitium is the space in between these renal tubules and vasa recta. These spaces are called medullary interstitium. The spaces in between the vasa recta, as well as the spaces between the loop of Henle. These potent spaces are actually called as what? Medullary interstitium. One of the peculiar feature of this medullary interstitium on going towards the inner medulla is there is an increased osmolarity from 300 to 1200. That is mentioned in your NCERT. So I have taken that particular point, 300 to 1200. Now you have to understand the main mechanism why these 300 to 200 conversion of medullary interstitial gradient is happening in our kidney. This gradient is actually made possible because of certain specific movement of, number one is NaCl and number two is urea. First let's see the movement of NaCl. It is very clearly visible here. I think it is very clearly visible here if you closely have a look here. You see here the NaCl is dropped in the you see here, there is our this is our ascending limb of loop of Henle. So this ascending limb of loop of Henle, see the NaCl is pushed outwards from this ascending limb actually to the interstitium, and you know here there is water molecule which is transferring to the outside, and you see this NaCl on reaching the interstitium is taken by whom? Is taken by our descending limb, of descending limb of vasa recta, and this descending limb of vasa recta actually transport this NaCl, and this will be reaching the ascending limb now, ascending limb of vasa recta now. So on reaching the ascending limb, they will be put back into the interstitium. So in effect, what is happening? Suppose this is our vasa recta, this is our Henle's loop. So Henle's loop, NaCl is actually transferred like this, and NaCl is actually shifted to interstitium by the ascending limb of Henle's loop. So NaCl is made possible in the interstitium. And from the interstitium, they are taken by whom? From the interstitium, they are taken by whom? They are taken by, again, this descending limb. This is the descending limb. They are taken by this descending limb. This descending limb take this and they transport into ascending limb, and this ascending limb again transport this. This ascending limb again transport this NaCl into the interstitium. In effect, NaCl is maintained in the interstitium in the maximum possible way. NaCl is made possible in the interstitium in the maximum possible way. That you have to. So the movement of NaCl, if I just summarize movement of NaCl that has given in your NCERT. So ascending limb of Henle's loop, from the ascending limb of Henle's loop, it is put to interstitium. From the interstitium, who is actually taking this? Is our descending limb of Henle's loop, descending limb of vasa recta, and descending limb of vasa recta transport this to ascending limb of vasa recta, and this actually reaches where? This actually reaches our interstitium again by this ascending limb of vasa recta. So if I want to draw that particular diagram for you, again I can draw. That is very simple now. Okay, fine. This is the way. So NaCl is actually transported like this, and you have to keep an important point. This NaCl which is transferred to this interstitial space, interstitium is the space between this. I have already told this interstitial space, this NaCl transport is actually actively happening. This is an active transport. So NaCl has transported like this to the interstitium. This NaCl from the interstitium is taken by whom? This is the descending limb of vasa recta. And this is the descending limb of whom? Henle's loop. This is the ascending limb of Henle's loop, and this is the ascending limb of vasa recta. So NaCl took this, NaCl transport this, and this will reaches the ascending limb. Ascending limb again pushes this into the interstitium. So NaCl is made possible in the maximum concentration in the interstitial space. That is the highlight. So you can just note that point as well. So NaCl is retained in interstitium. Interstitium means interstitial space by this kind of movement. Hope you got an idea now. So that is the story of movement of what? That is the story of the movement of your NaCl. The story of movement of NaCl. Now you want to know the story of movement of what? So NaCl we finished. Now we have to know the movement of urea. You see, again coming back to this particular diagram, you see the urea is transported by whom? Which is this portion? Hope you know this is the collecting duct which extends from cortex to inner medulla. That is shown here. This collecting duct in the terminal portion transport this urea into interstitium, urea reaching the interstitium, but this urea is taken by whom? See, this is the ascending limb of loop of Henle, but the peculiar feature of this particular part of the ascending limb of loop of Henle is it is the thin portion, as I already told. So the thin portion took this urea, and this urea is transported like this, urea transported like this, and again reaching the again urea is reaching here, and majority of urea, I think you know, will be passed through the urine, but certain urea is taken back to here into the interstitial space, and again urea is also retaining in the interstitium. Urea is also retaining in the interstitium. So I can just note that point as well. So movement of urea, if I want to summarize, it is reaching the thin ascending limb of Henle's loop. Ascending limb of Henle's loop in case of juxtamedullary nephron has two portions, thin ascending limb and a thick ascending limb. So this urea is actually reaching the thin ascending limb. It is not reaching the thick ascending limb. Thick ascending limb won't permit the entry of urea into it. It is only entering into the thin ascending limb mainly as per your NCERT. So thin ascending limb of Henle's loop, urea is reaching there, and from this it is reaching the collecting duct. Collecting duct also called collecting tubule. From collecting tubule it is reaching where? It is reaching our interstitium. It is reaching our interstitium. From this interstitium certain urea again taken like this way. In effect urea is also made more in the interstitium or retained more in the interstitium. So you can just note that point. Urea is retained in interstitium by this arrangement. So my dear students, what is the net effect? Then, or we can net effect we can just note like this. It is if suppose this is the interstitium, just imagine this is the interstitium, the net interstitium I just drawn like this, just a representation of the interstitium. So into the interstitium is like this. Interstitium, inside this interstitium much electrolytes are retained by this particular arrangement. So this the peculiar feature of this interstitium is now what you know is this is hyperosmotic, or this is having high osmolarity. As I explained the concept of osmolarity earlier, this is hyperosmotic medullary interstitium. How this hyperosmotic medullary interstitium is being made, I just explained now. Now you have to understand one important point. This interstitium, it is because of this interstitium water molecules are escaping into this from the collecting duct. Hope you know the movement of water molecules will happen from low osmolarity to high osmolarity, or simply water molecule reaches into the high osmolarity situation or high osmolarity place. So this medullary interstitium is a high osmolarity place. So into this space actually from the collecting duct water molecule reaches. That is also given in this particular diagram. See water molecule is reaching the medullary interstitium. Why water molecule is reaching the medullary interstitium? That we explained. It is because of the generation of hyperosmotic medullary interstitium. So what I have explained is simply written here. Both this movement means movement of urea, movement of NaCl and movement of urea. Both these leads to retaining of electrolytes in the interstitium in the interstitial space. Much suppose this is interstitium, again I am drawing. Much electrolytes are made possible here to generate a hyperosmotic medullary interstitium. Presence of such interstitial gradient helps an easy passage of water from the collecting duct, thereby concentrating urine. So suppose this is the collecting duct. Collecting duct is the place where collecting duct actually collects the whole filtrate or whole urine. So from this actually we can our water molecule can escape into this. This water molecule, if reaching the interstitium, can be absorbed into our blood. Usually it is absorbed into our blood. From here it is absorbed into our blood. So the blood content of our body will be increased, or the urine will be highly concentrated. We can pass a concentrated urine. That is the way how we will concentrate our urine. So the peculiar feature of our human kidneys are that they can produce, or our human kidney can produce, nearly 4 times concentrated urine than the initial filtrate form. That is very nice, right? Because the initial filtrate, as which is shown, again going back to your diagram, initial filtrate, see, this is the initial filtrate formed. This particular thing is this round is the initial filtrate formed. This is only having 300, but we can make up to 1200. We can make up to 1200 actually. So 300 up to it is according to the need of our body. When we drink lot of water, we will be passing dilute urine. When we are trapped in a drought situation like a desert environment, we will be passing a concentrated urine. We can adjust that according to the need of our body based on the presence of another hormone, that is our anti-diuretic hormone. I will be explaining the influence of ADH also, because ADH has got an influence to concentrate or dilute our urine. That is according to the need of our body. So I hope you got a picture how actually this medullary, hyperosmotic medullary interstitium is made. The whole story is lying in this hyperosmotic medullary interstitium, and into this hyperosmotic medullary interstitium, simply the water molecule from the collecting duct are being taken. Water molecule into the collecting duct, water molecule from the collecting duct are reaching into this interstitium, from where actually our blood is taking this water alone. From there, this water is being retained or taken into the blood. That is the way how you have to learn this concept. Then that will be very simple for you. Students, I want to make you clear another thing, because the difference in the osmolarity at different parts of the renal tubules or the PCT. At PCT it is 300. On reaching the bottom portion of the loop of Henle, see, it is 1200, and it reaches about 200 at DCT. So here at PCT, what actually is coming? PCT actually what is coming? The filtrate which has just filtered out from the glomerulus is coming. Hope you know the filtration at the level of glomerulus, we usually call it as ultrafiltration. Everything from the plasma, just except the plasma proteins, everything is just filtered out. So obviously that filtrate will be having the exact osmolarity of the plasma. That is, the osmolarity of plasma is around 300. That osmolarity will be there for the filtrate also at PCT. And on reaching downwards, hope you know the peculiar feature of this. What is this? Descending limb of loop of Henle is permeable to water, so water molecules are escaping. So on reaching downwards, it will get concentrated. So going downwards, the descending limb, it get concentrated, and it reaches up to what? Up to 1200. But what happens on going upwards? Going upwards, many NaCl is actually passing out, and that NaCl passing out is active transport. This is actually passive transport. This H2 passing is actually passive transport. So that NaCl is actually actively transported out. So NaCl means what is that? That is a solute is actually passing out into the interstitium. So obviously the osmolarity will be decreasing. So going upwards in the ascending limb of Henle's loop, we are saying about Henle's loop, it gets diluted, and up to 200. So at DCT, it is actually, usually it is isotonic. Why I am explaining this is because everything has asked for your NEET MCQs. At DCT, this is actually hypotonic. Why? Because it is 200. Usually happens at collecting duct. Collecting duct. A collecting duct and DCT is actually a point of difference. It is according to the need of the body, and also depends on ADH, anti-diuretic hormone. We will be explaining that. This collecting duct usually the urine will be concentrated. So on going downwards the collecting duct, urine becomes hypertonic. That is a fact. Okay, these things you have to keep in mind, but usually this collecting duct and point at the DCT level, that can vary a lot according to the need of the body, that we will explain. Okay students, so we have discussed the mechanism of concentration of urine. So we have told like we usually passes a concentrated urine under normal situation, but the actual story is the concentration of urine actually varies. It can be more concentrated or it can be dilute as well. It actually depends on the usually on the water content which is present in our body, as well as even our climate. Outside climate can even influence the concentration of urine. So what all situation? It is mainly based on particular hormone which is called as, what do you know, which is called as ADH, or anti-diuretic hormone. You will be learning about this hormone, the details on this in the chapter chemical coordination and integration. Now we have to check the influence of ADH on this particular concentration. So under let's see two scenarios. Let's see first for example summer season, or if suppose a situation like we are trapped in a desert, and we have no water. Okay, we have decreased water intake is there. So decreased water intake. So this situation, hope you know, this situation prompt our body to preserve maximum water inside our blood. So that is the situation. In this situation, you know, there is usually the increased temperature, we can say increased temperature. Okay, or there is increased water loss, we can say increased water loss is there. Here there should be what? Here there should be increased water reabsorption should be there. So what usually happens is increased ADH release. This ADH actually helps for absorption of water from the collecting duct and the distal convoluted tubule. You will be learning that. Okay, so there is increased ADH, so there is increased water reabsorption. From what? From the urine or from the filtrate. So we will be passing a concentrated urine. Okay, this thing should be clear in your mind. So now we can just imagine just the opposite situation. Suppose there is a winter season, and there is, or we can imagine like an increased water intake. We are drinking lot of water. Both situations are similar actually in case of this urine concentration. Or you can say there is decreased temperature, right? So decreased temperature means decreased ADH release. Decreased ADH release means decreased water reabsorption. Decreased water reabsorption means we will be passing more dilute urine we will be passing. So this is the way actually our body adjusts with the water content or the outside climate situation. So usually we passes a concentrated urine, but that can vary according to the water content of our body or the climate and all. It is mainly made possible because of a particular hormone, that is anti-diuretic hormone, that is usually released by our posterior pituitary under the influence of hypothalamus. You will be learning that mechanism in the chemical coordination. So hope you got the idea of the influence of ADH on the concentration or dilution of urine. So if you have got some, or if you have built some concepts on the mechanism of concentration of urine, let's do some MCQs related to this particular topic, which is mainly asked in the recent NEET entrance. Okay, so first question, which of the following factors is responsible for the formation of concentrated urine. So concentrated urine mechanism we have finished just now. It is given first option, low levels of ADH. Is it low levels of ADH? It is high levels of ADH. Increased ADH is actually promoting increased reabsorption of water, so concentrated urine will be passing. So that is obviously a false statement. Maintenance of hyperosmolarity towards the inner medullary interstitium, that is obviously the true statement, because we know the medullary interstitium is made in the hyperosmotic state, and that is the way into that hyperosmotic medullary interstitium our water molecule escape from the collecting duct, and this water molecule we are reabsorbed into blood. That's the way how we are passing a concentrated urine. So secretion of erythropoietin from that is not at all related to this. Okay, hydrostatic pressure during glomerular filtration, that also not at all related to this. So which one of the following is correct with respect to kidney function regulation? Okay, first option, when someone drinks a lot of water, this is also asked for the NEET entrance. When someone drinks a lot of water, ADH release is suppressed. Okay, when someone drinks lot of water content is actually there, sufficient water content is there, so ADH is not necessary. ADH actually promotes the water reabsorption. So obviously that is the true statement. So we can just go into the next. If you got a true statement, you just don't mark the OMR sheet like this. You just make sure that other statements are incorrect. You have to go through the other statements as well. Exposure to cold temperature stimulate ADH release. Never. Exposure to cold temperature inhibit the ADH release. We have already told that. An increase in glomerular blood flow stimulates the formation of angiotensin. This is the RAS mechanism we have told. It is not the increase, it is usually the decrease in glomerular blood flow which stimulates the JG cells to release renin. In affects the angiotensinogen conversion, etc. we have mentioned. So it is usually the decreased. So during summer, when body loses lot of water by evaporation, release of ADH is suppressed. Is it suppressed? When we are losing more water, our body needs to preserve more water. How we will be preserving? We will be reabsorbing more water from the collecting duct by increasing the ADH release. So it is not suppressed, it is favored. So obviously that is a false statement. So the correct statement is A. Now which of the following statement is true? That is again urine is hypertonic in DCT. We have told in we have just so it is 200 with respect to 300 in the plasma. So it is not hypertonic, it is actually hypotonic. That is a false statement. When the urine passes into the collecting tubule, it becomes hypotonic. Usually it is hypertonic, because we are passing a concentrated urine, and from the collecting duct we are actually passing water molecule outwards. So it is becoming more hypertonic when passing downwards. So that is a false statement. Urine is isotonic in the PCT. That is true statement, because in PCT it is the same concentration of the same osmolarity is maintained at the PCT. So it is a true statement. Urine becomes more and more hypotonic as it passes through the Henle's loop. That is a confusing question, right? Because Henle's loop, you consider Henle's loop, we cannot say like as it passes downwards it first becomes what? Hypertonic, and then as it passes upwards it becomes hypotonic. We can say that way. So that is not a complete statement. Also we can consider it as a false, because urine become more and more hypertonic first and hypotonic then. That is the way you have to say. So obviously the C only is correct. So that is this thing. Hope thereby we finishes the mechanism of concentration of urine. So this is the way how you have to practice MCQ. First make your theory strong by reading the NCERT, and then you do the MCQs and practice for your NEET entrance. Okay, thank you.